we'll have a very interesting talk, I think. It's a talk about communication systems in living organisms. Uh, because it will be given by Professor Irena Roterman-Konieczna, uh, who is head of Department of Bioinformatics and Telemedicine at the Collegium Medicum of Jagiellonian University. Well, it is something from the beginning of her official biography, but I think that the most important is that Professor Roterman is, well, it comes always with some, let's say, at the first sight, strange ideas, but then they are quite interesting, and to some extent, I think they maybe using the word revolutionary will be too much, but almost revolutionary will be just okay. So we had the privilege to collaborate with Professor Irena Roterman for something like 15 years, I think, in some European project, without projects, and it is important for Sano that Professor Roterman is a member of Sano International Scientific Committee. Very important body is Sano. So Irena, floor is yours.
So thank you very much, and thank you for introduction. And I will present not, of course, not the communication system in living organism as it is in its complex structure. I will just show one of the, I think, interesting part of it. So I will start with the very elementary definition. What is one binary digit? Uh, to my experience, I realized that the students of computer science, if I ask them what is the definition of binary unit digit, to my surprise, not many students know the correct definition. This is the selection of one from two equally probable solutions. Everything becomes clear when we take a look on the universal definition, which says that the amount of information, it is I, is equal to minus logarithm with the base 2 from probability of the event. And now, why the biology shall know this definition? Because this is very important due to um, our most important molecule, which is the DNA. And as everybody knows, everything starts with DNA, whatever the organism is. But we are focused today on a human organism. So the question is, how many bits are coded in the human DNA? And the answer, if we take a look on the definition, is very, very simple because in the DNA we have 4 different nucleotides. So every nucleotide is carrying exactly 2 bytes because this red P is of course equal 1 over 4. And the question is, so how much of information is carried by a human DNA? And this the answer is that this is about 1 gigabyte. And today, the gigabyte, this is almost nothing. This is very, very, very low amount of information. I don't think that even a pen drive of the 1 gigabyte is available in a shop. And it, the next step is that since we have 1 gigabyte in the input and what we get in the output, we must realize that everybody who is listening to me at the time of t equals 0, everybody of us was just 1 gigabyte of information. And at the output, we have the most complicated system ever found on our planet, which is the Homo sapiens. This is the absolutely highest complexity of the system which we can see in our surroundings. And now the question, the conclusion is that this example teaches us that this is the business is not a large amount of information to get a very complex level on the output. The point is that the clue of this relation is information processing. So the question is how this 1 gigabyte of information is consumed to produce the human body functioning on this high level of complexity. So the next conclusion is that we shall not look for extending the databases. We shall focus on how to consume even small amount of information to get the output of the highly developed level. And what is this consumption of the information? So whatever we study, biochemistry, anatomy, histology, or cell biology, or many other bio disciplines, we just study what are the mechanisms of this very poor amount of information consumption to produce this very complex system. And what I am going to show here is one of the very, very small part of the technique of the system which is functioning in a human organism. So far, calculating the amount of information in DNA, we were dealing with the elementary event. And now we go further to discuss the more complex event. What I mean the complex event? For example, the problem is that I want to reach a particular very well-defined goal. To win, I shall know what is the probability to win. I shall know what is the probability to reach particular goal, and this is symbolized by this P capital. And to make it very, very, very easy, I am using the very simple example. This is lottery. If we want to win in a lottery, the probability that I select the proper numbers is extremely, extremely low. It depends, of course, of the rules of lotto game, but anyway, usually this probability of winning in this sort of game is very, very low. So what we do if we wanna win, we just take a decision to participate in many attempts. So the P small is very small value. P capital, this is the probability that I win. And K, this is the number of attempts. So actually this is the number of coupons I am fulfilling. So the more coupons I fulfill, the higher is the probability of winning. Everything is clear when I take a look on the equation. So P capital, this is the probability of reaching a goal. P on the right side, this is the probability of the elementary event when I um, fulfill just one coupon, and K value, this is the um, number of attempts I in a lottery, the number of coupons I fulfill. And this is the solution, which is of course commonly known. For example, from I went from the lottery, which is not a good, not the pedagogical example, to even worse one, to war, because when we take a look on the movies on Second World War, we see the weapons, and we see also the factories producing many, many, many bullets, because the probability that I shoot just one and catch the airplane of the enemy is, of course, very low also. And to make this example more close to the nature, because we are going to talk about living organisms, we see our beautiful, wonderful plants, which during the evolution, the evolution discovered that the key way of reaching the goal, which means initiate the next generation, was used by evolution. And we may see this ballots, this coupons in a lotto game of plants which are producing many, 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 many seeds to reach the goal, to find the proper place to raise the new generation of the plant. And if we go even further, and if we go closer to biology, the question is how we, the human being, how we get the children. We get the children exactly the same way. The key way, it means increasing the number of attempts is the way to reach the goal. It means to have a child. Here I draw a very simple picture of sperm. We see that the number of seeds is usually very large, and they are performing sort of Brownian movement. And but the expectation of the member to get the new generation is that making this key number big enough, large enough, we can get the new generation. And from the cellular level, we go even more further to molecular level. Molecular level, actually, this is the very, very important subject right now. This is the immunological system. So when the living organism is born, the living organism is equipped with all processes which are necessary to survive. I mean, when we see this newborn child, we see that all physiological processes are passing perfectly well. But the one thing which is unknown to living organisms is a completely unknown external world. I mean, the newborn child doesn't know what the enemy is going to attack this organism. I mean, what bacteria are in a newborn surrounding, and what bacteria is going to attack this organism. So what the human body does is synthesizing so-called immunoglobulins, the molecules, which are responsible to recognize my own cells and the cell of enemy, I mean this bacteria. Every cell has so-called markers, and these turquoise symbols on the right side of this picture shows, visualizes the possible markers on the surface, and of course our cells are also equipped with a specific surface. So if we say, who are we? Usually we can say that our brain is saying that I am myself, and this is not true. The only system in the living organism is the immunological one, which is able to recognize own cells, because immunological system recognizes the markers on my own cells. So if the immunological systems recognize the foreign marker on the bacteria surface, um, sort of emergency is started. It means that the immunological system is ready to recognize this enemy markers. And now, since we don't know what what bacteria is going to attack us, so the immunological system must be ready, must be equipped with many, many different, if I say many different, I am talking about large number of K, the different molecules which are here on the right side in black color, molecules which have a complementary, may say, apparatus or something, also marker, which is able to recognize what cell we are dealing with. And now, since we don't know which bacteria is going to attack us, so the human body is synthesizing these molecules which are shown here in black on the right picture with different parts, these lower black fragments, and you must see that the upper part, the black fragment of the molecule, is always the same. So here is a very, very important moment, because everybody knows that the mutation is a very negative event. If the mutation takes place, some very uncomfortable situation may happen to the human body. So evolution elaborated a very complicated system how to protect all processes where the mutation can appear. Evolution elaborated even a system that when the mutation happens, for example, during the synthesis of a new DNA molecule, it is able even correct such mutation. So this was a very, very big effort of evolution to protect organism against mutation. And what happens here? And this we show on the left lower picture. So the black part of the molecule is always the same. It means the sequence, there are no mutation in this black part of proteins on the right upper picture. But for the chain fragment, which is responsible for recognition of the marker of the foreign cell, must be different. So, coming back to the lower picture, this dashed line shows us the DNA or mRNA sequence, which shall be kept unchanged. But certain fragments of the polypeptide chain in this black molecule shall be different. So, especially for synthesis of these black molecules on right side, evolution elaborated the completely opposite system to promote mutation, to get different markers, because it would be useless to synthesize the recognition system which recognize the same object. So going to the lowest level of organization of organism, which is the molecular level, the key way of reaching a goal, it means protecting organism against many unpredictable bacteria. The key way is the best one. It is like we can fulfill many lotto coupons with different combinations, counting that just one will be the proper one. The same is here. The different combination of synthesis of the markers in immunoglobulin molecule, which recognizes the foreign cell, also shall be as big as possible. Okay, and so I think that the biology and evolution really knew the equation I showed you on the former slide. But the key solution, the key-based solution, has its own characteristic. It means it requires a lot of energy, because if I must buy 1,000,000 coupons on a lotto game, or I must produce thousands of for the weapon, so it consumes a lot of energy. So if I select the key-based way, I must be ready to consume a lot of energy, and the immunological system consumes a lot of energy producing many different molecules, which, as long as we are not attacked by illness causing bacteria, they are useless. Okay, and now if we take a look on this equation, we see that the P capital on the left side, it depends also on the P small value. And now what is this P small value? How can I manage, how can I influence the P value to get the P capital higher? This solution, when I wanna make the p-value higher, requires not energy, requires requires information. What I mean saying requires information. Say that I come back to lotto game. I wanna increase the p-value. So what I am doing, of course, this is the action which is illegal. I enter during the night before the game is running, and I select some balls, and for example, I magnetize them, and I put the magnets also on this machine which is selecting the numbers. And in this situation, I can fulfill just one coupon for the lotto game, but before I can use this method, I shall recognize what is the system of selecting the balls during the lotto game. The next example, this war example. Once again, instead of producing hundreds of ballots for the war, we just produced one target object defined ballot, which is also characterized by this that this ballet is equipped with information concerning what is the object this particular ballet shall destroy. And continuing the former examples, we come to the plants. So besides the producing many seeds, some plants synthesize and develop rhizomes. What is rhizome? This is a very developed machinery. All the processes, physiological processes, are passing in this part of the plant, and this is something what is developed in particular direction. If the direction is not correct, I mean the surrounding is not good enough for this particular plant, the direction can be changed. So when we take such a plant from the soil out, we can see that these rhizomes, they are changing direction, looking for the best environment. And once again, we treat this method of getting a new generation in plants as a P way, because this part, this rhizome, is actually testing, so it has the information which place is good. The seed, which is carried by wind, falls down, having no idea whether the place is good or not. And rhizome is the organization which is selecting the best place to grow the new generation of plants. And of course, when we go further for cellular level, once again, I showed the sperma picture as an example for key way-based method to get new generation. We, the human, we have another method now, which is in vitro technique. And once again, we got information, we recognize the mechanism of this process, and in the in vitro technique, we don't deal with hundreds of sperms. We just take 4 or 5, and this is enough, because the information allows us to focus on the mechanism, on a process to get the success. And next one, which is the molecular level, when we use the vaccination, when we use the P-way. The P-way to get to reach a goal is a vaccination. So what is vaccination? We just push the immunological system not to actually not beside the random synthesis of the immunoglobulins carrying the markers. We want to be sure to get the one against particular disease, which is very important and very dangerous for us. So what we do, we just use the technique to make the system, the synthesis of the immunoglobulin molecules, to produce the markers which are shown here on the right side as a red dot, against this particular disease, this particular bacteria or virus, which is very dangerous for us. So, and when it was possible after recognition of the system of immunological defense against the sickness causing bacteria. So once again, without information about the system which is which is responsible for particular process, which I'm calling a goal, to reach a goal. So before I am able to increase this p-value, I must recognize the system I am dealing with. And now the next question. Aha. So once again, this vaccination. So we should get information how the system is working. And I put this information in the system to be sure that among all keys, there is the one which I wanna my organism to have it in the whole spectrum of these molecules. And next question, how do I recognize? I take a biochemistry manual, which is huge, very thick, very heavy, and a lot of information. So how do I recognize which process is solved, which goal is reached by the living organism on a key way or P way, particularly when I go to the molecular layer. So the way to recognize the P-based process is very, very simple. When I take a look on a protein molecule, you know that the whole body is just water and protein. And I see that the molecule, they call it receptor, is built by 4 different subunits, say A, B, C, D. And there must be a peptide coordinated. And there is a prosthetic group. It means that this the molecule, the organic molecule, which is not built on an amino acid. Sometimes some ions must be present in a certain points of certain places of the molecule. So what are all these ingredients? These are the parts of information, because if just one subunit, the D, doesn't appear for some reasons, who knows why, it doesn't appear, the whole system doesn't function. And it means that the D part must be present in all this construction to make the system complete, to make the information coded in the system complete. And now, if we go to a very, very simple protein, this is the lysozyme. This is the enzyme, the turquoise part, this is the protein, and I distinguish here these 2 yellow amino acids. So, as I said, this is enzyme. The information in this enzyme is of course present in these 2 yellow parts, but not only. If we take just these 2 yellow parts by themselves and nothing else, it doesn't work. The whole other part of this protein is ensuring the very special, very specific environment. This is like an external force field for the reaction which is passing between these 2 yellow parts of this molecule. So here, looking at this molecule, I instantly see how much of information is coded in this one object. But as I said, this is just the simplest enzyme in human organism. If we go further, we can deal with huge molecules. We call some of the proteins, sometimes even we call them machines. For example, this protein shown here from two different views of the same protein. This protein is constructed by 21 amino acid chains, which are distinguished on by colors. And you must know that this protein, anytime it is synthesized in our organism, adopts exactly the same structure. So the P, this P information is coded in this particular construction. Even if we go further, for example, this one is once again the molecule constructed by many chains, and this one is the example of receptor. This is the part which can be recognized by, for example, immunological system, or this is the receptor which is working on a surface of the cellular membrane just to recognize whether the information can enter to the inside part of the cell or not. So this is highly, I would say, high level of information. So once again, if I see the process in which this particular protein is participating, I know this is a P-based way of communication. And even more complicated system, this protein molecule is actually very hard to show because this is a huge one, and this is the ribosome. This is the system, the construction responsible for synthesis of proteins. And if we can put all proteins, for example, taken from the biochemistry book, if we want to put them in a ranking order, I think that the ribosome shall be on the top of this list. This is built by many protein and RNA molecules, and every part of this molecule is carrying particular part of the information, other words, is responsible for this increase of P, this small p value, to make the P capital higher. Yeah, and the process of synthesizing this ribosome, actually the lowest picture on the right side, this is the huge, also we call it machine, huge protein and RNA construction, which is participating in a protein synthesis. And why this ribosome is on the top? As I said, this is the almost most complicated system on a scale of protein construction. But we learn also from this top position that the process which is accompanied by this very complicated molecule is also the most important one. And on the other side, we have also here a system of control. What I mean control, as I said, with this hypothetical molecule at the beginning of the discussion about proteins. So if one piece, one part of this huge construction is not present, the process doesn't start. It means it shows on the same time that the synthesis of protein is a very highly controlled process. And really, If you take a look on a cell, the most dangerous event, which is actually destroying the whole cell, is the uncontrolled synthesis of the proteins. If in a cell the synthesis of the protein is random, this is the biggest disaster for the living cell. So the more complicated structure of the protein participating in the process, the higher P value is for reaching a goal, which is, for example, in this case, the production of the protein. Generally speaking, the students of medicine or whatever the life science see the word specificity in biochemistry manual, they shall be aware that the word specificity, it means higher P value for particular process. Yeah, and of course we can calculate a very, very simple simulation how the P capital get changed, how P capital increases for K and P small value. So when we take into account that we see that the key increase of key makes a very fast increase of P capital value. But we must keep in mind that key way, this is the energy consuming way. On the right side, we have a change of P capital depending on P small. So it is not so rapid like on the left side, but in the solution on the right side, we must be focused. How do I get information about the system to modify its functioning? Yeah. And now when we come back to the problem of 1 GB, which is to me, for example, this is terribly small amount of information, particularly in respect to the high complexity of the output product. So I may recommend a very simple publication. Data are given here. This is quite old already, but I think this is very important and related to the subject I tried to discuss today. So what the authors say in this particular paper, they are using cancer as an example. And what is the problem? Almost every hospital has the laboratory of cancer research. And the authors claim that the databases concerning cancer are huge. If we take all the databases, even those available in internet, so this is a huge amount of data about this disease. And they ask the question, what for we do this experimental research on cancer? They claim that if we close all cancer laboratories right now, we don't lose anything, because they claim that we shall focus our attention on transformation of this information we already have. Information about cancer, I think, is bigger than 1 GB. So I truly recommend this paper because this is a very, very interesting approach. They even claim that the opening of the new discipline called uh, mat- uh, mathematical cancer, discipline called mathematical cancer should be erected in education. Yeah, the conclusion from their paper is that we are in the opposite situation. We have huge data and very, very little of interpretation. And to make this discussion complete, I shall recollect this motto. There is nothing more practical than a good theory. As you may see, many authors are given as, many people are given as the authors of this sentence, but I think that this is still very, very open and actual opinion. I think that all the listeners are my friends. So before I say many thanks for your attention, I would like to show you the book. We presented our vision of functioning of living organisms, and this is just a very small part of the global view of living organisms. Thank you very much.
Thank you very much. And well, we have time for question and for discussion about this concept. So what you have presented, it is quite good high-level description what is going on in our live organism. To which it is description from high level. To which extent do you think that it will may be useful to predict behavior? You have shown an example of such a prediction, but I think that
Yeah,
trouble is going into more details. Do you think that it will be good,
yeah,
appropriate for this?
yeah. So this is a very good question, and I shall say that at the beginning of the presentation. So the model I presented is just the molecular level. So we don't discuss a very important system present in human organism, which is the nervous system. The neurological system is organized completely using completely different methods. This is the word of electric signals. So the behavior and the whole mental word activity of the human, this is not the object of our discussion. The model I presented is focused on the chemical molecular part of our body.
So it is just one aspect.
Yeah,
Yes,
yeah.
thank you.